This course is a course on biological network dynamics. And I typically have taught it at a fairly elementary level because it's normally a mixture of undergraduates and graduate students. And the mathematical background for students tends to be not that strong in this department. And so back when this course was offered in physics department, which it was for many years, we did a little bit more in terms of dynamical systems theory and stability theory. And that's something I can certainly cover in this class if you're interested. But fundamentally, the issue is going to be how networks work in biological systems. And those networks can become very, very complex, although here we're going to, at least typically in the context of this course, we tend to stick to fairly elementary concepts. The course really is going to talk about modeling dynamics on networks. I often will talk a little bit about how you build networks because these days, modern molecular biology gives you pathways, it gives you connections, but it doesn't really tell you about how to model those things kinetically. And there are some simple rules of thumb you can apply that give you reasonable guesses about what the model structures are, which can be pretty useful. And then we like to think about how to analyze these networks in terms of dynamical systems, and then how they work in the context of especially multicellular signaling, which is something that I'm personally quite interested in. And again, when I've taught this course in the past, we've had a pretty diverse audience. Alex, who's on the call here, is now up in Indianapolis, but he's an experimental biologist. We've had pure mathematicians, we've had chemists, and so on. And so it's always a little bit different depending on people's backgrounds. We do rather different things. So the basic concept that this class treats is a network, and networks can be a very general organizational concept. And there's, of course, a whole institute of network science here at IU in informatics. But they tend to be focused on the structure of networks, on the architecture of networks. And this course is going to be focusing on their biological deployment, but the methods are pretty general. You can use them in a variety of areas. So if we think about networks as an abstraction, I still have that United Airlines, that ancient United Airlines diagram. I hate that example. I need to replace it. But airline route map would be a classic network. Of course, we have things like the World Wide Web, interconnects between hubs, the famous protein interaction network from 25 years ago. And then here at IU, we have people like Kati Berner who are very interested in others, Kyle Sterling and others who are very interested in things like citation networks and how scientific ideas overlap. We also have things like trees, which are an example of a network, world trade networks for economics, human disease networks. In this case, that human disease network is actually talking about not infection, which we'll come to in a minute, but rather the relationship to different kinds of diseases. The World Trade Network, in a sense, is a dynamic network, because you could think of fluxes on the network, but these other networks are more static in their behaviors. We have real neural networks in your brain, and you also, of course, have artificial neural networks, which are typically semi-dynamic. Normally, you run a neural network until it reaches a steady state. Although if you do things like recurrent neural networks, you can also have dynamics in the neural network. And then you have networks that are intrinsically dynamic, like infectious disease networks, where you have somebody who coughs on somebody else, and so there's temporal ordering involved in the network behavior. When we come to biology, and here I'm using the word biology to mean really what happens inside the body, because ecology is also a kind of biology, but of different scale, has some of the same issues, some different. We find that there are four main categories of network that we want to address. What are called metabolic networks and chemical reaction networks, that are the things that create and destroy the components in a cell, handle energy transfer. That's what Alex and I were chatting about a little bit just a minute ago. We have signaling networks that relay information within the cell, typically from the cell membrane into the cell, but also within the cell from region to region. We have gene regulatory networks that turn genes on and off in response to the state of the cell. And then mathematically very similar, although functionally very different, you have what are called physiologically based pharmaco-kinetic networks that essentially represent the transport of material in the body, in particular blood, sometimes also lymph. And those are very important if you want to do pharmacology. So these days, when you design a new drug and you want to get the drug approved by the FDA, you have to be able to show in both in computer simulation, when you take this pill, how is the chemical active ingredient absorbed, usually in the gut? How is it distributed in the body to different organ systems? How is it metabolized? How is it broken down? And then how is it eliminated, excreted? It's so-called ADME, absorption, dosimetry, metabolism, and excretion. And that PBPK models can become very, very complex. But in fact, since everybody has the same set of organs and the same blood supply, the PBPK models actually all are pretty similar. You can take those boxes and open them up and make them more elaborate, but for the most part, they're pretty well established. And if you look, if you're interested in the industrial side of things, by far the biggest use of this kind of network modeling in industrial is in the PBPK space. These days, pharma companies can't get enough people with those kind of credentials because they have to have them for their FDA certifications. So those are a couple of the kinds of network structures that we have. Again, mathematically, we're going to find that we treat them all pretty much the same. And certainly if you come from a physics background, you'll have seen many times that rather different systems, physical systems, can wind up giving you the same set of ordinary differential equations or partial differential equations. You can get the same math out of different things. And so the general mathematical tools that we're going to develop, computational tools that we're going to develop, are going to be pretty applicable in any of these contexts. There also are other kinds of networks that maybe are a little bit, all of those really connect directly to molecular species, to chemicals. If we talk about population dynamics, this is getting more in the realm of ecological description of networks. Here we're talking about populations of cells within a tissue, but we could also be talking about populations of animals in an ecosystem as well. And those have a typical structure where you have replication of individuals, death of individuals, and then differentiation or migration of individuals. Now, in a real tissue, and this is also true of all of the systems that we're looking at here, there are spatial structures, and networks typically don't consider those spatial aspects, although they can be extended to do some spatial elements. The last kind of network, which has become pretty fashionable these days in the context of medical digital twins, and also in the concept in toxicology of what are called outcome networks or disease networks or adverse outcome pathways, where the nodes represent some kind of behavioral state of the system or some set of transitions that occur going between, for example, here, you're exposing a tissue to a particular, in this case, toxic chemical. And that toxic chemical initially will bind to molecules within the cell. Those cells will then do things and lead to a cascade of what are called outcomes or adverse outcomes that eventually lead to an illness or damage. And so those are the basic network classes that we're talking about. This is typically a class that draws a little bit from a variety of disciplines. We have a little bit of biology, some biochemistry, some certainly of bioengineering and biophysics, definitely a little bit of computing, although not terribly sophisticated. At least we don't require it to be very sophisticated. The data science can be very helpful if we want to be able to map and understand networks. Dynamical systems theory and some basic mathematics we need. And if you're going to do things like fitting parameters to experimental data, then you need to have some basic statistical understanding as well. Typically, and you may be one of the exceptions to that, most people don't come into a class like this having had all of these things. And so it's useful to remember that. Again, if we have an N of 1, maybe that's a little bit of a funny thing to say. But historically, when we had a big class, we would try to pair people up. So if we had somebody who was a wet lab biologist and somebody who had done a lot of computing, we might team them up. And again, you can tell me what your background is and how it works with this. This is officially in an engineering program, and science is really about prediction. Can we build models that are able to predict what we observe? And how do we discover those models? Engineering is a bit more on how we apply those principles and those models in particular to control systems. And while this class typically doesn't get to the level of sophistication of really trying to build control systems with digital twins, that's certainly the area that I'm interested personally in going in. And I think it's the direction that biomedicine and biomedical engineering are going. There are a lot of problems with doing that. It's not so easy to actually build predictive models of people. And we can talk about that perhaps as we go forward. So what are we trying to do scientifically? Well, we have some understanding of basic biology, and we'd like to be able to observe, unlike physics, where you have a sense that there is in fact some underlying first principles reality. In biology, we're always phenomenological. We never have completely detailed models. And so at some level, it's surprising that you can make quantitative predictions at all in biology. Especially the kind of network models here, we're making a lot of very, very strong simplifying assumptions. And it's a little bit unexpected, perhaps, that you can actually be quite predictive about many things. We do a lot of what I'd call post-diction, which is you observe something and then you want to replicate it. So you're trying to build something that mechanistically is able to reproduce something you've already observed. It's more interesting if you could actually predict something that hasn't been observed yet. That can happen. It's not always possible, but it can happen. And ideally, you want to couple these things back to experiment, either in best case to suggest new experiments, certainly to interpret experiments. And there are a lot of situations in which it's difficult to control or understand experiments, or the experiments are quite expensive. Or if it's in a human, you can't do the experiment at all because of ethical reasons. And so then having computational models is really a big help. Something that's become more relevant, I think, in the past couple of years with the boom in machine learning, artificial intelligence, is that when you're using real world data, especially in biomedicine, you face two fundamental problems, or at least two. One of them is that there is no gold standard annotation in biomedicine. If you have, say, pathology slides from a cancer, you have some pathologist who's looked at them and then said, this one is grade 4 and this one is grade 3. And you can certainly build a classifier, a machine learning classifier, image analysis classifier, that will go in and tell you that if I show it a slide, this is grade 4, grade 3. But to the extent that the person doing the classification made mistakes, the system has learned those mistakes. The same thing for things like image segmentation. And so with unsupervised learning, you could do a little bit better than that, perhaps. That's an area of interest today. But one of the things that people have done a lot of is build what are called synthetic data sets. If I build a computer simulation and I know exactly what went into that computer simulation, then I have gold standard data. I know what the parameters were that generated the simulation. And therefore, I can test whether my machine learning actually is able to recover those parameters. Now, again, that's not typically something I would do in this class, but it's something that we could do. I work with several PhD students on that topic at the moment, and it's an area of interest to me. One of my former students just published a paper a few weeks ago on that in the context of cancer diagnostics. So not doing the thing I teach in the spring rather than in the fall, which is he builds agent-based models of tumors. You can then control the parameters in those agent-based models to simulate early stage, mid stage, and late stage tumors. You can then simulate what would happen if you did a biopsy and made a histology slide, and then you can trade off of that. And that works pretty well. These days, to be honest, the image classification systems from, say, Google or App or Facebook are so good that people typically do some kind of transfer learning where they use the generic classifier, and then they do a second stage of trained classification off of the latent space representation that the initial classifier gives you. And we're actually working with a master's student at the moment who's just starting a project on that. But that's one direction one can go with this. Another direction you can go is that, again, in the real world, by definition, you don't have what are called contrafactuals. If something can't exist in the real world, you can't create it as a part of your training set. And so if you have situations that are very rare but significant, or things that you want to avoid happening, it's difficult to put those into machine learning contexts without doing synthetic data. And so there's a nice paper by Mark Alber and a variety of others from Marcus from UC Riverside where this is a couple of years old now, 4 or 5 years old now, where they were talking about how to integrate this kind of mechanistic modeling with machine learning. And actually, in October, there'll be a big conference at NIH on how to integrate mechanistic modeling with machine learning effectively in the context of digital twins. Again, this course tends to be really on the mechanistic side, but it's interesting to combine these things with machine learning when one has the right people. Okay. So again, coming back to why this is an engineering course, fundamentally, if we want to do successful engineering, we need to be able to design things. And if we're going to design things, we have to be able to predict. If we can't predict the effect of what we're going to do, then we can't design. We can do things by you can do combinatoric exploration. Goodness knows a lot of things were developed by chance, but it's typically more efficient if you can actually predict what's going to happen. And in particular, if you're in medicine, biomedicine, you have a current state of the system that you're looking at, a health state. You have some desired output state, say getting over the cancer, for example. And you have some set of manipulations, for example, drug therapies, chemotherapies, radiation, and so on. And so you'd like to be able to design interventions that get you to the outcome that you want. And I should say that medicine today certainly follows that paradigm, but it follows that paradigm in a fairly loose way. It's not very automated. And so design is something that really is not implemented very much in many areas of contemporary medicine. There definitely are areas of contemporary medicine that are designed. If you have a surgical implant, that will be designed to fit your body. A cancer radiation therapy will be designed for the particular tumor that you have. But if you go to the doctor with a cold, well, cold, he won't probably do anything for you, but you go to the doctor with a bacterial infection, he'll give you some standard course of antibiotics and say, come back in 3 weeks and tell me whether you got better. And that's not really design. And certainly one of the things we'd like to be able to do, and a place this kind of modeling is used rather a lot, is in trying to identify drug targets. Some place that it's being used more, although not as much perhaps as it could be, is in optimizing drug dosing schedules, for example, for cancer. Synthetic biology, can we design circuits, biological circuits that do particular things that we want to do? And biochemical engineering is a whole area where chemical reactions are designed as well. So that was sort of the background here. Again, it feels a little bit odd to talk to one person about it, but I guess it's okay. We're supposed to be meeting Tuesdays 4:10 to 6:40 here. Sometimes we'll probably be online. If you're sick, or I'm sick, or I'm traveling, or you're out of town because you're at a meeting, we can do it online. And I've always tried to be flexible about that. I think there's definitely advantages to being in person, but it's not always possible to do that. The textbook for the course is this Herbert Sauer Systems Biology Introduction to Pathway Modeling. That's a pretty elementary textbook, but it's I think it's quite a nice one to begin with. There are a lot of other textbooks that are written in a similar way, either at the same level. There's also a nice online course from Caltech that covers some of this material that I should probably add a link to. There's Eberhard Voigt's first course in systems biology, takes a rather different perspective. There are a bunch of, if you look at online, there you'll find plenty of these courses. Herbert's textbook you can download for $8, as a PDF. If you go to his text, if you go to his page here, you can download the PDF for $8. So I would recommend that. And Herbert, who is a long-term collaborator of mine, has written a number of textbooks. The one that we're using is the second one here. The third one here is more on the mathematics of dynamical systems. The fourth one, an introduction to metabolic control analysis, digs into the biology a little bit more. It's a little bit sort of a second semester type course. And the enzyme kinetics also is a little bit more chemical reactions one. They're all good. They're all well worth reading. But here's the $7.95 PDF. So, and actually, this particular textbook, we've been using it for many years, and so we've contributed. This class, when it had a lot of students in it, basically beta tested that textbook. Almost every page in it was rewritten, and every exercise was redone based on the feedback from this class. So I'm not an author on the textbook, but I feel some sense of contribution to it. Yeah, well, I have a credit in it in the acknowledgments, and the class has a credit too. The other thing that Herbert has, which are quite useful if you're interested in digging it a little bit deeper, is there's this YouTube channel, Center for Reproducible Modeling, by BioME here. And they have a seminar series and also a course series, which has, here's their 2024. So they teach a short version of this course, where I sometimes lecture, actually. And which covers a lot of this material. And they also have, yeah, that's me there. Oops, I'm not screen sharing the right thing. Sorry about that. I apologize. This is something that I have to say. So the textbook is here. If you're interested in sort of understanding how these kinds of things can be used in a very wide range, right? Gene expression modeling, protein signaling modeling. Uri Alon has a great textbook on this topic from Israel, from the Weizmann. Heart modeling, population dynamics. Remember, we were talking about the different kinds of network models there are. So he has a series of lectures here that cover all sorts of different applications of these kinds of metabolic modeling. So if you're interested in learning more about these specific topics, he's got a great set of lectures here. There's Paul also from IU, Paul Backlund, one of our faculty there, giving a lecture on the kinds of multi-scale modeling that Paul and I do. Good lecture on parameter estimation there. So again, it's not an expectation that you will necessarily use these additional resources, but there's quite a bit out there if you're interested, depending on the direction that you want to look at. Yeah, those kind of biomechanical models get to be very, very elaborate. I mean, my own research tends to be more biomechanical. We do agent-based modeling of multicellular organisms. This is more basic, perhaps, but it's a necessary part of understanding these more complex models. This course, again, if we have one person in the course, it's a little bit odd to have it structured this way. We can reorganize it, but typically there's a weekly homework, or some of the homeworks last 2 weeks. And this year I was thinking of doing a midterm test, a brief test. And then typically it's mainly a project course, And so the goal is to find something that people are interested in and work that through as a little project. Nothing dramatically ambitious, but something that's interesting to them. Well, that's a very interesting question. I mean, that's something where I would do better on a blackboard probably than I would on a slide deck, which I have prepared. There are a lot of and it's a question that's more. The question was essentially for Alex and Hayden, who may not have heard. The question was really, what are the bottlenecks, computational bottlenecks in this field? Where do we need the most assistance in terms of accelerating and making practical computing? And so, again, given who normally shows up for this class, because the level is usually fairly elementary, we tend not to focus on those kind of areas. But those are very important areas if you're actually going to take these things and apply them. So one of the things that's expensive is parameterization. If you have experimental data set or synthetic data set and you want to reproduce that data set with a model, there are a series of tasks in terms of optimization and validation that need to be done. And even very simple models could be surprisingly hard to parameterize. In other words, if I give you, at the end of today, let's see how far we get, but at the end of today, we'll write an exceedingly simple model in the software tool that we're going to use. And if I gave you a series of time series from that model and the model structure, and I asked you to give me the parameters that were used to generate a specific time series, well, you could use SciPy optimizer functions to do that. They'll take quite a while. They typically don't give you very clean results. If I start adding noise to that system and I ask how does your ability to infer the parameters go, the answer is it degrades very rapidly. And so this is not just a so back in the years when we had a lot of people take this class for things like 2020, 2021, when we taught this class around modeling infectious diseases. And back in 2020, at the beginning of the COVID epidemic, people really wanted to know, for example, how many people are likely to get COVID and how many people are likely to die. And they're a very simple network model, which we could run today. We're not going to do that one, but we'll do an analogy to it, which is called the SIR model, susceptible, infectious, recovered, or /dead model, where people are uninfected, they're exposed to somebody who's infected, they get sick, they pass the infection on to other people, then they either recover or die. In the case of COVID, of course, you get it again, but that wasn't part of the original understanding. And that model doesn't have that many parameters in it. But what you would like is to know what those parameter values are so that you could say, gee, this is how many people are going to get sick, how many people are going to die. And that you're watching this infection take off and you want to fit that infection curve to the real world data sets and infer what the parameters are. And people's estimates differed by factors of 50 for those parameters. Now, of course, as time went on, the fits got better. But early on, it was very hard to get even rough estimates of what those parameter values were. A second problem that you face besides incomplete data, which was certainly true of COVID, is that the parameters don't stay the same. Once people realized that they could get COVID, they started doing things like social distancing, wearing masks, and so on. And so the infectivity of the virus effectively went down. And now that nobody wears masks and things anymore, it's gone back up. And so how do you fit time varying parameters? It's not trivial to do. So those are two places which actually require a fair amount of work. And there's some nice software written by Denise Kirschner at University of Michigan called Calipro that does Bayesian statistical analysis to try to do that kind of fitting. And it's fairly expensive. You have to run, the individual simulations are not expensive. Running one of these simulations takes a few seconds of computer time. They're not, I mean, for the simple ones that we're going to do here. But to do parameter estimation, uncertainty quantification, you typically have to do tens of thousands or 100,000s of replicates of the simulation. And if you really want to do control, you want to be able to say, what do I have to do to be able to get the outcome I want, given my initial state? Well, the space of perturbations that I can apply is very, very large. And so now the dimensionality of the problem gets to be very, very high. And so then you're running really lots of replicates. And so that's an issue. Another place where you could wind up having issues is typically, again, in the simulations, say that SIR model for COVID really only has 3 or 4 parameters. But when you get to more elaborate network models, you can have dozens and dozens of parameters. And so there's no way you could do an exhaustive search. 10 values each of 100 parameters, you have 10 to the 100th possibilities. You're never going to be able to do it. And so there's a lot of interest these days in machine learning and smart optimization. I should say I'm not an expert on that, but it's an area I'm interested in exploring too. And you can certainly begin to apply it in these simpler contexts as well. Of course, if you had good high qubit quantum computing, it would solve your problems because you could explore the outcome from the simulation with every possible parameter set. If you had 100 qubits and they had enough resolution, well, you could simultaneously explore 10 to the 100th possibilities, and then you would be able to do this more easily. But you would still, even then, face the problem of incomplete data and noisy data. And so this is a big problem when you want to do human medical digital twins, because the classic example of a medical digital twin is an airplane turbine, jet engine. But jet engines are kitted out with sensors that report thousands of times a minute about dozens and dozens and dozens of different states. We were just talking about, there was a question about sort of what the choke points, it's not what was on the slides here, what the computational choke points are for realizing these kinds of models. And I was trying to explain some of the places. I think, again, this course itself doesn't do much usually in the context of machine learning, although it could. But some of the places where there are serious technical obstacles to realizing the vision of what would be called medical digital twins, the ability to actually predict how human health states evolve and control that. That's the direction the world wants to go very much. And I think everybody in this field would like to see us go there. Probably that's something for your generation more than mine. In other words, we're the people in my world are trying to set up the problem so that they're going to be solved in the next 25 or 30 years. Maybe sooner than that, given how good large language models have gotten. They maybe solved sooner than that, but we'll see. All right. So I was just talking a little bit about recommended reading and background. The thing that you missed that probably is relevant was screen share the other picture. Okay. It was simply the textbook, if you decide to go ahead with this, the textbook, I keep, unfortunately, what I see and what I see here on my screen and what's there are not the same. So the textbook is downloadable for $8 from this website. So if you oops, wrong. If you're interested, don't buy it from Amazon for $50. Go to Herbert's website and download it for $7. Okay. So that's all I wanted to say about that. All right, come back to the screenshot. So, and as far as course logistics goes, I was saying we're going to do things primarily in Colab. That'll be convenient if you want to try to hook it up to machine learning models. But you could certainly download the software. Is quite simple, but if you could certainly download it to local computer, run it locally. And in the past we used an online server called NanoHub, but NanoHub's gotten a little bit old-fashioned, and running in Colab has a great advantage that if I create exercises and things, I could just email them to you and you can run them that way. Yes, so Tellurium, so that was the sort of the next thing I was going to say. So Tellurium is a Python library, and Herbert, if you know Herbert Sauro, who's the one who wrote, actually a big part of it was written here at IU by Andy Sumoji, who used to be a associate instructor in computer science, although he's not here at the university anymore. But actually the back-end solvers and things were all written here at IU quite a few years ago. But Tellurium is a Python library, and it implements a very simple but quite effective model specification language called Antimony. And one of Herbert's Sauro's quirks, if you like, is that every time he does a project, he gives it a new name. And so every piece of software he's written in every language he's written have new names. And so in any case, Tellurium and Antimony Tellurium is the Python library that implements Antimony. All right. Another thing that you might want to look at, especially if you're a little bit more sophisticated about these things, is this biomodels database. And let me screen share that for you. The biomodels database is a repository of several thousand validated models. Well, 1,073 validated models and 1,592 unvalidated models, which represent a whole variety of interesting biological networks. And these are expressed in a language, a language called Systems Biology Markup Language, SBML. But SBML has the nice feature that it can be translated back and forth with a single command into the Antimony that we'll use. SBML is what's called an exchange language. Markup languages are not generally very human readable. You can read them if you have to, but they're not much fun. And so Antimony basically is a human readable version of SBML, which is why we're programming it. But I definitely recommend browsing the biomodels database for ideas. So, as I said, we need to know a little bit about a lot of things. It's nice to know a lot about a lot of things, but you don't have to know a lot about a lot of things to get started. And so I often say with this kind of modeling, fundamentally, you need basically a Wikipedia level knowledge of a bunch of different things. And so for things that you may not have run into before, you might start with that and then fill in and deepen your knowledge depending on where you are. Again, for historically people who take this class, the mathematical background is often not as strong. And so that's sometimes the place that people need to work. So we're going to begin with the beginning of Herbert's textbook. So this lecture, next lecture, chapter 1, and then we move from there. And then there's a general appendix I on modeling. Herbert also has really good online documentation. And so for Tellurium, there's this Tellurium Read the Docs. It has great exercises and basics. And I have a handout, which is one of the things I really like about Tellurium, we'll come back to that in a little bit, is that it could do a lot, but it's an extremely simple language. And so basically the whole language fits on two sides of a piece of paper. And so that to me, in terms of learning, is very productive. You can certainly do the same things with say Mathematica, and you could do a lot more with Mathematica too, but it's also, there's a bit more of a learning barrier to people to do that. And of course, the fact that it's a Python library makes it convenient because you could use it in conjunction with whatever Python you like. If you run it in Colab, then you could use it in conjunction with whatever network software you like to use with Colab. Just some general terminology, and again, if this may be very familiar to people here, it may be a little bit artificial. The concept of a model as an abstraction, and it may seem unnecessary to say this, but I'll tell you, when you work with people who come from very different fields, the terminology is often very different. And so a model takes some set of input parameters in what I call a model structure. You might say mathematical structure if you like, although it doesn't have to be mathematical. It gives you some set of things that in principle are measurable. And that seems pretty trivial, but you might be surprised how often even people who are apparently very sophisticated write models that, for example, produce things that could never be measured or depend on parameters that could never be determined. And so to remember the purpose of a model is actually pretty important. And if you look in for example, what the Food and Drug Administration is interested in, or the European Medical Association is interested in, reliability, verifiability, and trust are things that are of very substantial interest. And so this idea of a model as something that takes a structure, some kind of computational structure, and some set of input parameters that gives something that's intrinsically measurable is really pretty fundamental. And as I said earlier, before you came in, one of the things you face in biology is that nothing is from first principles. Everything is phenomenological. And therefore, fundamentally, you never expect your models to be perfect. You're always going to expect them to be only partly accurate. In addition, real biological systems are highly stochastic. And we will talk a little bit about stochasticity later, but we have to accept the fact that if you could somehow redo your life twice, you would be different. Networks as a concept are pretty simple. We have a concept of a node. These are graphical models. They're not spatial. A node and some kind of interconnections or links, and there's some state that lives on the node. And if you're a real classical network scientist, what you care about is the architecture of the node. But we're going to be focusing more on dynamics. And so here we've got something on the bottom where we might have chemical reaction where A, B, and C come together to produce D and E, or A, B, and C come together to produce D. And so these links can be thought of as as just relationships, for example, publication relationships, or they can have a temporal component too, which they will in our world. And typically, if we're doing science, what we want to do is first infer what the network that describes the system that we're interested in is. And then we want to be able to simulate it and understand its potential behaviors. And so we're really in science of network modeling is about really reverse engineering. When we're doing engineering, we tend to be going in the opposite direction. We start with a network, and then we go forward from there. The key thing about networks is that they're a pretty harsh abstraction of reality. In particular, they leave out space. And space is often very important. Two things that could react chemically with each other don't react if they don't come in contact with each other. And so networks tend to leave out the context, which can be a limitation. Although it's possible to build network models that sort of simulate spatial relationships. As an example, we were talking earlier about these pharmaco-kinetic models, where you have models of the blood and the distribution of chemical species in the blood. But these days, people often will build a model of the gut, where the gut box is broken down into subsections. And so you have an idea of, you take a pill and the pill is in your throat, and then it's at the top of your gut, and then it's passed down through the intestines and so on. And you build a box for each one. So you have a cascade which simulates the spatial organization of the gut. And the reason that you would do that is that the pH, so the acidity and the temperature and the absorbance of the walls of the gut differ at different positions. And so when you take a pill, the place in which particular molecules in a medication are absorbed is different depending on where it is in the gut. And that can actually be used in a productive way, because you could design carriers for medicines which release their cargo in the right place to get the maximum dose into the tissue that you care about. So it's possible to build quasi-spatial models with networks. I should say that normally we have the idea of the nodes as the things that are being modeled, and their states living on the nodes. Generally, we don't build substructure in the nodes, although it's possible to do that. Economists, for some reason, like to go the other way, which is economists like to have the nodes be the relationships and the links be the things that are being related, so they flip it. But the key thing is that we're typically going to have a subset of dynamics where links represent interactions. And the key thing is that an interaction is going to change some aspect of node state. There are a lot of different notations that are used. Arrows are the ones that we're going to run into, and you'll see a lot of different formalisms for arrows in biochemical networks. There are there's the old joke that the great thing about standards is there's so many to choose from. You'll find that arrow diagrams are often not standardized. One of the things I was saying, the textbook is one that was beta tested by the people in this class over many years. The early versions of this textbook, almost every diagram in the textbook had a different convention for how the arrows were used. And now I think after quite a few years, I think there may be some inconsistencies, but we have tried to make sure that the arrows are used consistently in that textbook. But in general, we're going to have a concept of transformation and a concept of regulation are going to be the two things that we're going to be worrying about. And here was my comment that some people like economists flip the net backwards. If we're talking about networks, fundamentally, this very simple concept of nodes and links can be very flexible. You could apply it in all sorts of different ways. You can have one type of node, or you can have multiple types of node. You can have one type of link or multiple types of links. The links can be bidirectional or unidirectional. And something we'll run into in biology a lot is that classically in a network, the links, the arrows only point to nodes. So links only connect nodes to each other. In biology, very often we'll have arrows that point to arrows, which are modifiers or regulators. And so that's a little bit different convention from what they use in other fields. We're going to be talking about network dynamics. People who do classic network science typically are mostly focused on the topology or structure of the networks. We are going to be focusing on how, and they're also interested in how those topologies and structures change in time. We're going to be focusing instead on dynamics on networks, where typically we're going to have a fixed network architecture, and we're going to see how the node state changes in time. Now, there are what are called adaptive networks, where you change the state of the node and the architecture of the node together. And in some ways, you can think of, for example, gene regulatory networks as functioning in that way. But typically, if you're going to be doing classic network theory, you'd be asking, say, if I add a web page to the internet, how many links is it going to have in 10 days from now? So that's changing the network architecture. In biology, for the most part, the network architectures are established, at least within the body, and therefore we're going to be looking more at how the dynamics play out within those networks. And so what the state is living on a node could vary depending on our context. It could be the fraction of the number of people who are of age such and such in the population. It could be the number of people who are susceptible versus the number of people who are currently infected with influenza in the population. It could be the concentration of a particular molecular species within a cell. It could be the fraction of a particular molecular species that are phosphorylated or active inside of a cell. It could be the firing rate of a neuron. In our case, almost anything that can be represented as a number, as a scalar, is fair game. Although, of course, network state could have a lot of different kinds of types as well. So this kind of basic concept, what we'll call reaction kinetics, which is essentially networks of first order ordinary differential equations, is used everything from neuroscience to economics to epidemiology to ecology and elsewhere. And so mathematically, almost all of these different systems and networks can be represented with the same relatively simple mathematics. And that means you can represent them using the modeling environment that we're going to be using. And so again, we're going to have a set of nodes which have states or values, links that define how those nodes' value states change in time. That is, we're going to have rates of change, rate laws, which exist at nodes due to their link interactions. And so what's going to be different in a different system will be the architecture, which nodes are connected to which other nodes, and the rate laws that operate in that architecture. And the first thing we're going to start with, and this is the basic set of concepts that we have in, maybe it's worth stopping for a minute since I've been talking for quite a while, and ask is that, I realize this is pretty elementary so far, but does that make any sense to people, or is this, uh, as I say, some years people have seen this all before, some years people have never seen it, and so I don't know where people are coming from. It's sorry, you're familiar?
Yeah, familiar.
Okay, so I can speed up, I guess. Have you both, I mean, so chemical reactions, I both done some chemistry. So basically, when you go to antimony tellurium, the basic concept that antimony and tellurium implement is a chemical reaction. And so you have a concept, for example, here, 2 molecules of ADP go reversibly to a molecule of ATP and a molecule of AMP. You put the chemistry, you put the substrates on the left, the products on the right, and you can write stacks of chemical reactions that sum. Chemical reaction networks conserve mass, although typically the biological networks that we're going to write don't respect mass conservation. And again, the mathematics is going to be a series of rate equations where each one of these arrow diagrams for chemical reaction implements one or more rate equations. So the nodes typically then are going to be chemical species or some abstraction thereof. Typically, we're going to think about their state as being the concentration of that chemical species, because usually the rate laws depend on the concentration. When we do things like transport networks, like pharmaco-kinetics, pharmacodynamics, then we usually use amounts rather than concentrations. And the basic arrow represents either transport or chemical transformation. And so anytime you see a pointed arrow, that's going to represent either the transport or transformation of one chemical species to another. And so a link is always associated with a rate of action, the rate at which that transformation happens. So when we're going to model chemical reactions, we have to come up with some rate laws. The simplest rate law that we're going to run into is going to be what's called mass action kinetics. And again, this is really just to get us through some very basic use of the software today. So we're going to start with the simplest possible chemical reaction. Well, I guess the second simplest. First, simplest possible chemical reaction might be A goes to B. The second simplest would be A plus B goes to C. So we'll imagine one molecule of A and one molecule of B to combine to produce one molecule of C. And we want to know what the rate of production of C is as a function of the amount of A and B. And typically we start out with the diagram, and we can talk in more sophisticated ways about how you can guess what the rate laws need to be, even if we don't know that a priori. But for here, we're going to go through some basic steps. The first thing that we're going to do is we're going to use concentrations. So we'll typically use the italic version of this. The species name will be ABC, and the italic of that will be its concentration. And then we'll write the rate of the reaction as the velocity, VF. A plus B goes to C at a rate VF. Now in chemistry, if this is a real chemical reaction, then that rate will depend on a lot of things. It'll depend on the pressure, the temperature, the pH, and so on. But one thing that we know in general is that if it's a forward reaction, it's possible that C has some effect on the rate of reaction, but in general, the rate of forward reaction depends only on the concentrations of A and B. So we expect that VF is a function of A and B. Okay. There already are a lot of assumptions in writing something that simple because chemical reactions actually are discrete events. They're not continuous. And so we have to assume that the molecular concentrations are big enough that we can write a concentration rather than count individual molecules, and that the rate of reaction is high enough that we don't have so much stochastic fluctuation that we have to worry about it. And we can deal with the stochasticity and counting of molecules within the same framework. And we're also going to assume that over whatever volume we're considering, the concentrations are effectively uniform. We're neglecting spatial variation. And so now we want to think about what this implies mathematically. Again, it's very simple, but it's a surprisingly powerful formalism. So even if it's very familiar, it's probably worth going through it at least once. So this chemical reaction, A plus B goes to C, implies 3, for doing continuum assumption, 3 first-order ordinary differential equations, the rate of change of A, the rate of change of B, and the rate of change of C. And they all depend on the same rate law, which is VF of A and B. And so one thing that we do have to get used to a little bit, if you're used to writing things out as ordinary differential equations, is that when you write rate laws, one arrow diagram like A plus B goes to C gives you 3 ODEs, one for each chemical species. But vice versa, there might be situations in which you need multiple arrow diagrams to implement one ODE. So it's not quite a one-to-one mapping there. And I should also say this is something that only becomes an issue maybe later on, is that the mapping from an arrow diagram to ODEs is unique. If I write the arrow diagram, I can always derive what the ODEs are. If I write the ODEs, I cannot uniquely go backwards to the arrow diagrams. And so you lose information when you go from the diagram to the mathematics. That was a point I did not appreciate when I started this business 30 years ago. Herbert Sauro was the one who really emphasized how important that is. So the question was, why is it that going from that you can't always go backwards from the ODEs to the arrow diagrams? And the answer is that I could show you multiple arrow diagrams that will give you the same set of ODEs. Now, you could say, well, if the math is the same, why do I care? But if I want to, for example, come up with a drug target where I'm going to add something to the system, I actually do have to know what the underlying structure of the system is when I add that drug target. And so the classic example, which I used to give as a homework assignment, I haven't done for a while, maybe it would be a good one, is John Tyson at Virginia Tech, a great mathematical biologist, and we'll do some of his models later on, one of the great experts in the modeling of the cell cycle. And I take one of his papers from 20 years ago, a very famous one, in which he is modeling how the cyclins in the cell control each other to build the cell cycle. And if you look at that paper, it has something like 14 ordinary differential equation rate equations. And I say, what are the signaling and reaction networks and gene regulatory networks that he is representing with these 14 ordinary differential equations? And if we did that as an in-class exercise, you'd look and say, that's easy. And then after half an hour, people will say, we can't figure it out. And the paper doesn't have a diagram. So you actually cannot determine from the paper what the network was that he was modeling. Now, in his case, there are plenty of other papers that he wrote where he does have the diagram, so you could find it, but you can't do it. And I mean, I don't want to lean on that too hard, but if you want things to be reproducible and you want to communicate, the networks are fundamental and the math is derived. I spend a lot of my time, and actually next week, actually Thursday, I'm heading to Germany for a meeting of an organization called Combine, which is Herbert Sauer is one of the founders of, which is an international society that builds standards. This is not probably not exciting to anybody in the room or on the call, but it builds standards for how to represent these things so that they're reproducible and interpretable. That if you're going to build a model for yourself that's never going to be used again, then it's fine to do whatever you want. But if you're in the world of engineering or the world of pharmaceuticals, it's really important that everybody understands what you've done and that they be able to extend it. And so coming up with standard notation and terminology it's very unglamorous. It's a little bit like sewage engineering, but it's also very important. This systems biology markup language standard that I referred to is a product of people working for 20 years, dozens and dozens and dozens of people working for 20 years to come up with something. If you look at it, you might say, well, why did it take so long? The answer was that they had to think very carefully about how to represent these concepts in a way that's reusable. Okay, so we have this concept of rate loss. And we have this rate where we've asserted that the rate of reaction depends on the concentration of the substrates. And now, if we want to go any further, we have to have a guess for what a rate law is. And the simplest guess that we have is what's called the law of mass action. And if you've taken chemistry, you probably start out with the mass action sometime in maybe the first week of your class or second week of your class. And you could even make a pure mathematical argument for a continuity argument for basically as a Taylor expansion. Mass action is the lowest order Taylor expansion of a generalized rate. That's a slide that I don't usually include because I try to generally in this class not put in as much math, but basically you notice that if you don't have any A, you can't have a reaction. You don't have any B, you can't have a reaction. So the rate of reaction has to be zero. If A, the concentration of A is zero, the concentration of B is zero. And the simplest way to get that is to multiply A with A to B. In fact, the lowest order term has to be A times B if you want that to work. And so the law of mass action says that the rate of reaction is linearly proportional to A and B with a rate constant K. Now that rate constant could depend on a lot of things which we're not modeling. Again, the temperature, the pressure, the pH, and so on. But given a fixed temperature, pressure, and pH, you expect the rate to be linearly proportional to A and B. And that's the law of mass action. You can justify the law of mass action on physical grounds. And if you look in a biophysics textbook like Phillips, it'll do that derivation out in detail. Fundamentally, the rate of reaction is the probability of having your individuals of the two molecular species close enough to each other in some finite volume. And if it's a one molecule of A reacting with one molecule of B, the probability of being within that volume is proportional to the concentration. And so the probability of having them both inside the volume is just the product of the two. And so now I can actually write the rate equations. If we want to now find out what the time evolution of A, B, and C are, we have to specify the initial conditions how much A, B, and C we got, or the initial concentrations. And then we also have one rate constant. So this one arrow diagram, A plus B goes to C, assuming mass action, tells us that we need 4 parameters to get the time evolution. We need K, the rate law, rate constant, A0, initial value of A, initial value of B, and initial value of C. Now, if we run that in time, these are concentrations, so they better be greater than or equal to 0, and they better stay greater than or equal to 0. Of course, if we're modeling other things, they don't have to be positive. And if we start out with some amount of A and B, and then A and B will decrease monotonically in time, and C will increase monotonically in time. And we could put some constraints on the system, simple mathematical constraints. If we start out with equal amounts of A and B, we'll use up all of our A and B and have no A and B left at the end. So our steady state will be no A and no B, everything turned to C. If we start out with unequal amounts of A and B, we'll have the reaction run until we run out of either A or B. And now we can actually simulate it. And so now, if you can, I would ask you to write a little simulation. And that was what I was hoping to do for the rest of this afternoon, if that's okay. And I'll show you the beginnings of this Antimony language. Everything we're going to do is going to be in Antimony and Python. Well, everything, I should say, everything we're going to do about the networks, quad networks, will be in Antimony and Python. You're free to use any Python libraries you like to wrap those. Antimony syntax is not quite Pythonic syntax, and so that's something one has to get used to. But it's extremely simple. I would recommend running Tellurium on Colab. The great thing about Colab is that it doesn't require you to install anything on your local computer. You can also go to the Tellurium site and download to your local computer a variety of IDEs, including Jupyter Notebooks, local Jupyter Notebooks if you want. If you have a Google account, of course, you already have Colab, so most people probably do. If you don't, you're going to need to access it. And as I say, if during class, even if you're in the room, I generally encourage people to run the Zoom with the sound off so that I can put things in the chat to share links and that kind of thing. It's easy to overwrite what you've done with Colab or to lose your files. It doesn't. It's the file handling is not as sophisticated as one might like. Otherwise, it's pretty effective. So if you're on the Zoom, you can take the notebook that I shared, which actually has the completed exercises, so it's a bit of a cheat. But I guess I don't have to tell people how Jupyter Notebook works here. But so we're in Python. We have interactive cells, shift enter to execute. When I share the notebooks, I'm going to share them in a read-only form. So you'll have to you have to copy it. So if you open it and then copy it, you can then make your local copy. Okay. And then you'll have to save it to some place that you have write access to. Actually, you can keep it for the moment if you're on that internal machine. You can keep it depending on how you logged in. If you have a Google Drive, you save it to your Google Drive. Otherwise, Colab will give you some scratch space to work with. Okay. Again, if anything is confusing or you want to screen share back to check something out, it'll be fine. You come back to me. I recommend having the Tellurium Antimony documentation open in another window on your desktop or laptop. This Tellurium Read the Docs is particularly useful. I've printed out, again, on that piece of paper, as I say, the whole Tellurium language, pretty much the Antimony language fits on a single piece of paper. I printed out a Python cheat sheet as well, in case you haven't run into Python much. We don't use very sophisticated Python. The first exercise was open this template notebook. Again, you'll have to save your code. I think actually what I shared with you, the notebook I shared with you has the completed code in it. This is essentially the completed exercise. To run Python, to run Tellurium in Colab, you will need to, one minor disadvantage with Colab is that every time that you reopen a session, you have to reinstall your libraries. Now for Tellurium, which is not a huge library, that's not a pain, not too much of a pain. When you do it for CompuCell, which is the software package that my group develops, CompuCell is several hundred megabytes, and so the install takes a little while. And so if you look here, this first cell here, you have this install libed curses 5, and then you have a pip install Tellurium. And all of this stuff here, the percent capture and the os.kill os get PID 9 is because Colab will throw a bunch of warnings and error messages, which actually don't prevent you from running Tellurium, but those are traps that hide those error messages so that when you run it, it looks clean. Okay. Does everybody have that? Do people need to, can people see it? It's pretty small. Do you want me to? So if you run this cell, nothing should happen. Of course, if you have a local version of this, then you don't have to worry about the install. Your session crashed, huh? Interesting. Well, let's see if I don't.
Just need not sure what the live in versus.
Let's see. See if that'll work. There are Python compatibility issues because of the way Google manages its Python library packages. And so that's what that one is trying to is a compatibility check. So we'll see if the rest of this works. Okay. So let us see if it works now. It does. So it looks to me like what has happened here is that Google has probably updated a library that was previously not compatible. And so that command is now not needed anymore. Let's see if this is okay still. See, does that work? So it's actually looks like it's the import os kill that's doing it anyway. That worked. Okay. So try it without the. It looks like the libcurses works, but it's the import os that's causing the problem. Okay, did it work for me? Again, we haven't actually done anything so far, so that's not the most exciting thing in the world. What I'd like you to do, if you look at your Tellurium notebook, there will be a little command here in the demos. If you go to Tellurium, read the docs. It is a quick start, and in the quick start there is some sample Tellurium code, and you paste it into the cell under the Tellurium load and run it. Okay, does that work? Okay, read the docs, quick start. Grab this, import Tellurium, etc. Come back. And now, I actually don't want to override this. I don't think, okay. Actually, it's already here, so paste that in, run it. And you should get this. Did that work? Alex, are you trying it? Did it work?
Yeah.
Did you have to delete those two lines did you have to kill those two lines 5 and 6 in the install?
Well, I actually tried because I couldn't remember how to do it because I'd started before and I was the Gemini, so I asked it how to do it. And it didn't have those to begin with.
Okay.
So I didn't have, but yeah, I had in the same, yes. I just didn't have them.
All right. And it didn't throw an error message when you
No.
did it. Yeah.
No.
You should have gotten something that looked like that, which is a plot of a chemical reaction. Now, this is an even simpler chemical reaction than the one that I wrote down to begin with, because it's just species 1 becoming species 2 at a rate proportional to species 1. So it's just exponential decay. But that's all that it is there. Okay. So let's just look at the antimony code. And antimony code is almost embarrassingly simple. The first thing that you'll notice is that antimony model specifications are not Python. It's a different language, and antimony is written as a string. So typically we'll use the one that's in the example that we pasted in is inline, so there are no carriage returns. But typically we'll put that string in triple quotes because then we want to be able to have carriage returns, and that's the Python convention for carriage returns. And so we will put everything, our models will be in the form of character strings. The good news about that is it's simple. The bad news is that when antimony crashes because of parse errors, it doesn't give you very informative information about where the error was. And so that means that debugging is a little bit of a pain. Ironically, there are two things that you'll run into, and I'll come back to this in a bit. One is that upper and lowercase are different, and if you have a lot of names like K, A, B, C, it's easy to accidentally type a lowercase a when you mean an uppercase a, and it's hard to see it when you've got a lot of them. And so upper versus lowercase is one that you get. The other one that you could get, which could be more of a bear to catch, is that if you give a reserved word, if you use a reserved word as a variable name or a parameter name, it doesn't tell you that, it just crashes. The one that usually people get stuck on is gamma. Gamma is the reserved word for the gamma function. And so if you want to call something like alpha or beta, I recommend putting a number after it so that you don't run into reserved words. Obviously, don't call a variable time. So if we're going to take what we have already and we'll write the reaction A plus B goes to C, again, we'll have this boilerplate code that first loads tellurium as TE. Then we will load A. TE.load A is the method that parses. It's a parser for the tellurium string, the antimony string. And then simulate is a method that runs the parsed model and will plot it. In this particular version of the code, we're not saving the intermediates except for the parsed model. Typically, we would put the output of simulate into an array, a NumPy, an array, so that we could manipulate it later on. All right. So we're going to write the chemical reaction A plus B goes to C, and what we're going to wind up with is going to be changing a couple of lines of the code that we have. And again, I realize this is very elementary, but we can move on from it pretty quickly. And you'll notice, we'll come back to this, but you'll notice if we look at A plus B goes to C here, what we're typing is almost identical, A plus B, arrow C, semicolon, rate. And that's really why I like Antimony so much, because you more or less type what you see. So the basics of Antimony syntax, there are essentially 5 concepts, which are all in that handout. Unlike Python, indentation and spaces do not matter. Interestingly, because Antimony is a declarative language rather than a procedural language like Python, in most cases, the order of the lines you type does not matter, which is something we're not typically used to in programming. That's a specific of declarative language. There are a couple of places where the order of the lines does matter, but in general, if you reverse the order of 2 lines of Antimony, it doesn't change the output. Capitalization does matter. If you're used to C or C++, semicolon is the separator. I recommend that you end every line with a semicolon. Carriage return will do it for you, but it's just better practice and sort of good hygiene to always use a semicolon at the end of your lines. And I apologize in advance if I sometimes leave a semicolon out. So we're going to write the chemical reaction. The first line, which we'll have to do once per simulation, will be to import our Tellurium library. Classically, just the way NumPy tends to get imported is NP. We tend to import Tellurium as TE. You're welcome to import Tellurium as Tellurium or something else if you prefer, but we typically use TE. And so all your methods you'll see will look like TE dot something. Then we're going to do our loading of our model. And so we'll have R equals T dot load A. So R is a handle to the parsed model. And again, we typically will use triple quotes. You could either use triple double quotes or triple single quotes, which allows us to have carriage returns inside of the string. I will point out that people probably have run into this before. If you use Word, because you want to say type an article or something, and you put the strings into Word, Word will happily change your quotation marks into different characters. And so you have to remember to undo that when you copy from Word. So it can be surprisingly difficult to catch that error when people do that. So I've mentioned it now. Of course, we can give the parsed model, any handle we want. And here I'm giving the literal string inside of the load A, but normally I would say something like model equals model string, and then I would say T dot load A of model. So I would have the string be a variable. One thing that you'll run into where things are less convenient in Tellurium is that if you want to change the model structure, say I want to say what would happen if I added a line, an extra reaction to my simulation. When SBML and Tellurium were written, people didn't really think about it that way, and so so you have to then do string operations. Grep awk or something else to do the string manipulation, because you actually have to edit the character strings. Something that has been implemented at our request is that you can actually make stoichiometry, which we haven't come to yet, variables. Now, there are a couple of little inconveniences that we'll run into, but for the moment, I don't think we'll have to do it. So you put everything in triple quotes. So the fundamental concept in Antimony is a chemical reaction, although we can write ODEs explicitly if we want to. So here in line 3, we have R1 colon. R1 is a name for the reaction. We don't need to name reactions, but if we do name the reaction, R1 will have the value K times A times B. So it makes the rate of reaction available to us. And we have A plus B dash caret C, and then a semicolon in the rate law. And that's all there is to it. One thing we do have to remember, and it's again easy to make a mistake when you're cutting and pasting, is you cannot have two rate laws with the same label. And so if I type R1 colon A plus B goes to C, and then a new line R1 colon C plus D, R1 again goes to something else, then that'll throw an error. So if you do name your reactions, you have to remember to give them unique names. So instead of what we had before, just type R1 colon A plus B goes to C, semicolon K times A times B. And here I imagine, as I said, I left off the semicolon at the end of the line. I should have had a semicolon. Now, if I type just R1 colon A plus B goes to C at a rate K times A times B, it will crash. And it's going to crash because Antimony and Tellurium expect you to define initial values for your variables and the parameter values. If you don't give it initial values for the variables, it will set them to 0. But if you have a parameter like K and you don't give it an initial value, that will crash. So I have to type R colon A plus B goes to C, semicolon rate K times A times B. I have to give initial concentrations for A, B, and C. Here I've set A to 10, B to 10, and C to 10. My exercise says to set A to 10, B to 10, and C to 0. Those are initial conditions. And again, if I hit simulate here, it's going to crash again because I don't have the value of K specified. And so the last thing is I need to specify the rate constant K. I just set K equal to 1. And so if you could type those four lines and run it, give it a try. Okay, so the next thing we have to do is we've loaded the model. Tellurium has two basic simulate methods. One is an ordinary differential equation simulator. The other one is a Gillespie stochastic simulator. And so I'm going to run the simulation, and the call to the simulator is initial time, end time, and then the number of substeps I'm going to use. So this is an explicit time stepping. It's not an adaptive time stepping. There are a variety of settings you could use to modify the simulator, depending on whether you're to stiff equations and whether you have various things going on. But the default one usually works pretty well. And result will be a Python object that will have at the top of each column the name of the column. The first column will be time, and then the second column will be species A, third column will be species B, fourth column will be species C. And so if I do r.simulate of 0, 50, 100, there are going to be 100 time steps, 0 to 99. And now we can run that. I don't need to tell you how to use the Jupyter Notebook, but if you forget, it's there. I usually get something that looks like that. The last thing that we need is we need to display the output. There's a built-in function in Tellurium called plot, which plots auto-scaled graph with a little key with the species in it. In general, that built-in plot isn't very flexible, and so we'll usually use matplotlib instead. But if we just want a quick and dirty plot, we don't want to remember matplotlib commands, r.plot works. All right. If we were to display a result, as I mentioned, you get an object and the object again has the time in the first column and then the values of A, B, and C in the remaining columns. The order in which the columns occur, time is always the first column. The identity of the other columns is not always in the same order, but they do have alphanumeric labels. So I can ask for the column A and do slicing that way. So it's actually a pretty nice object. The default that I gave you here, simulation time of 0 to 50 is too long. Everything is over. And so we have to usually come up to begin with by hand, or we can analytically determine what the time range should be. Here, 0 to 10 with 100 time steps is pretty good to improve what we see. So we get something like that. And that's the basics of the method. If you've got that running, you could try changing K. And of course, as you change K, the rate of reaction will change. And so effectively you're rescaling the x-axis of the simulation. So if you do that, here I have K of 0.01, 0.11, 10, so on. So it's a chemical reaction. The rate is going to depend on A and B. As I mentioned, with a simple reaction like this, the reaction will stop when we use up all of whichever species we have less of. And so it's interesting to change the amounts of A and B, the initial amounts of A and B, and see what happens. If we start out with no A, for example, we start out with A equals 0, then whatever amount of B we started with just stays there because there's no A to react. So if I started out with C equals 0, I'd have nothing happen. And so here I could take the initial values of A and B to be 1, 3, 5, 10, and 15, for example. Again, what I've written here on the right was just the base code that we started with. I should have probably changed C to be 0 in the example. And you can get something that looks like this. So the reaction will proceed until you've used up all of A, if you have more B than A, or all of B if you've got more A than B. And so this is all pretty simple, but you're already getting things that aren't totally trivial from even the simple chemical reaction. We can get very complicated very fast if we want to. Or better get the syntax down before we start playing too much. Okay, so why don't you try then taking the pattern that we've just started with and now rewrite this so that you have what's called a cascade reaction or sequential reaction. A goes to B at a rate Ka times A, Kb, B goes to C at a rate Kb times B, C goes to D at a rate Kc times D, times C, Kc times C. Start out with 10 units of A, none of B, C, or D, and initially make Ka, Kb, and Kc equal to 1. And it's up to you whether you name the 3 reactions or not. Something that I did not do, which I will do now. Did this work? Did this little exercise work? So you should have gotten something that looked like that, right? When you make K, Kb, Kc equal to 1, it's a fairly trivial result. If you now make the powers, make Ka, Kb, and Kc different, things get a little bit more interesting. It's a little bit more interesting if you have more stacks. So here, when you only have 4 reactions, A to B, B to C, C to D, you don't see it as clearly. But if you say had 6 of them, A goes to B, B goes to C, C goes to D, D goes to E, E goes to F, F goes to G. And then you try Ka's 1 KB is 10, KC is 100, would be 1,000 and so on. Or vice versa, 100, 110, 1, 2, 1, or make them powers of 2. It gets a little bit more interesting. One thing that you need to know is that exponentiation in antimony is a caret. So the exponentiation symbol in Python is a double star. It's an up caret in antimony. But if you try this out, these two paths, you'll see you get quite different trajectories of your time evolution. You can get one in which essentially all the reactions happen at the same time and the amplitude goes down. And the other one, all the reactions basically have the same amplitude, but the time gets stretched out exponentially. So even this very simple cascade reaction can actually give you some non-trivial temporal and spatial and amplitude behaviors. The last thing that I had here was to add one to look. What we had here was we start with A, everything goes into D. And so at the end, we have a chemical equilibrium where we have no A, no B, no C, and all of our A's become D. The other one that's more interesting is if we now couple that loop back and we say D goes back to A at a rate KD times D. And now the question is, what changes when we do that? How is our final result different? And if people can try that out, again, if you've done chemistry, this will be something you already know, and you may even have done this exact simulation either mathematically or computationally. But it's worth asking the question: how are these different? Because in real biological systems, we deal sometimes with open systems, like the one we started with. And sometimes we deal with reaction loops like this. And so it's useful to already begin to think about how those differ in what they do. If you want to try that, I could give you a minute to do that. So you have to add one line, essentially one line here below that, and one new rate constant. This was what we started with. So if I close the loop, and the rate constants are all the same, I wind up with equal amounts of A, B, C, and D. And if I break that loop, which I could do without having to edit all the reactions just by making the return rate 0 here, wind up with everything going into D. So in fact, the time evolution of these is quite different. In one case, I wind up with equal amounts of A, B, C, and D. In the other case, I wind up with all D. Now, if you remember doing chemical equilibria, if I made the reaction rates different, then that will control the equilibration ratio. So if I make KD, for example, 2, let's make it 2 instead of 1. I now wind up with half as much D. So the equilibrium amount is inversely proportional to the rate. So that's the basics of Tellurium. That's all I had prepared for today. We could build up quite quickly from here to do rather elaborate biochemical reactions. And we can also use the functionality that's built into NumPy and SciPy to do things like look at stability and bifurcations of dynamical systems using this kind of methodology. But again, today the goal was simply to get everything running. And as we found out, the standard install sequence that we had working until this morning didn't work anymore. So there always tend to be a little bit of unexpected results. The other thing that I was hoping to do more this year would be to look at some of the published models, of which there are, as I mentioned, just in that biomodels repository, 1,000 of them. Find models that are of interest to people and begin to ask questions about, for example, how do you do parameterization and uncertainty quantification? So something that could be of interest as well.